pretty much all we did, right? We talked a little bit about enzymes, so there's some enzyme stuff in there. We did a whole lab on enzymes too. But we talked about enzymes, right, just because cellular respiration, right, is a process that has lots of enzymes. And photosynthesis is another process that has lots of enzymes. Right? What are the 3 parts of cellular respiration? What does cellular respiration begin with? Did you take glycolysis? Yeah, you're going to have to yell your answers because I can't hear you just as much as you can't hear me. This is really important. What comes after glycolysis? Sure, which is kind of a linker step too. What comes after glycolysis? Well, this is really basic. We're not even into the actual exam stuff yet. What comes after glycolysis, y'all? There we go, the citric acid cycle. What comes after that? Yeah, otherwise known as? Very good. What about photosynthesis? What comes first? What's that? The light-dependent reaction, right? And then after the light-dependent reaction is the light-independent reaction, which is also called the Calvin cycle. Is there an electron transport chain in photosynthesis? There is. Is it in the light-independent or the light-dependent part? Light-dependent part. Very good. Okay, so those are the basics, right? Those are the 2 basic processes we talked about. Okay, catabolic versus anabolic. What does catabolic mean? Catabolic is to break something down, right? Who can name a catabolic process that we've studied in this unit? Basically all of cellular respiration, right? Glycolysis is breaking down glucose, right? What's anabolic? That's building up, right? Photosynthesis is an anabolic process because we create something out of substrates, right? Versus cellular respiration, which is breaking that back down to those substrates. Forms of energy, we talked about very briefly. What are some forms of energy? Yeah, okay, those are all good ones, right? Chemical energy, right? An important one for the process we talked about. Light energy, very important, right? A byproduct of those processes sometimes is heat energy, right? And within those we have potential versus kinetic, right? A big example of that is the flow of protons down their concentration gradient through the ATP synthase to make ATP, right? That is the conversion, right, of potential, right, energy to kinetic energy as those protons flow down their concentration gradient. Let's see here. How could we apply the laws of thermodynamics? What is the first law of thermodynamics? That's what it's called, but what does it mean? Explain it. Yeah, right. The amount of energy that goes into the system, right, is the amount of energy. Right. It's we're not going to get more energy. We're not going to lose any of that energy. Right. It might change forms. Right. The energy often changes forms. Right. But the amount of energy is constant. What's the second law of thermodynamics? I'm assuming you said that as we convert from one form of energy to another, right, it's an inefficient process. Is that what you said? I cannot hear you back there. And this is really terrible. I'm going to hate this. I can tell you right now. Yeah, right, so when we do all those conversions, right, the energy is neither created nor destroyed, right, but it does change forms, and when it does change forms, right, we often get a loss from the pathway. Right? This is where body heat comes from, for example, right, as we change from light energy, right, to chemical energy, right, we're going to lose some of that energy. Right? Light bulb, common example. Right? If we put electrical energy into the light bulb, right, we get light energy from that, but we also get an amount of heat energy from that. Let's apply that to a picture. Here we go. Do we see the first law of thermodynamics or the second law of thermodynamics represented here? I mean, really perhaps both, but the second one more obviously, right? Where do we see the second law of thermodynamics represented? Yeah. Right here. Right, so to combine these two things, right, requires 3.4 kcals per mole. When we hydrolyze ATP, we get 7.3 kcals per mole. That's more than we needed, right? So there's a net difference of 3.9. We had 3.9 extra kcals per mole. Where did those 3.9 kcals per mole go? Probably heat, right? We use the amount of that 7.3 we need to do the reaction. The remaining part, right, probably lost as heat. So that's an example of the second law of thermodynamics, right? That it's an inefficient conversion, right? It's kind of like trying to pay, I think the example I used before, trying to pay with everything with $10 bills, right? In a system that doesn't give change back, right? So you need to buy an item that's $3, you have a $10 bill, you just paid $10 for it. Where's that other $7 go? Who knows? Somewhere else, right? That's what this is representing here, right? We had more energy than we needed for that particular process, right? And so the rest is lost as some other form of energy. What about the term inorganic? What does that mean? Yeah, inorganic, right, means the reaction consumes energy, right? Exergonic means the reaction gives off energy. Which one is spontaneous? Yeah, why? It's a hard question to answer the way that I phrased it. Right? Exergonic reactions are spontaneous, right, because they contribute to what? The what energy of the universe? Yeah, the free energy of the universe, or entropy, right? So exergonic reactions, right, are those that release energy back into the universe, right? That's what entropy is, right? Entropy is the tendency, right, of things to destabilize, to fall apart, to release that energy that was holding them together back out into the universe, right? So exergonic processes are spontaneous, right? If it's inorganic, right, we have to put energy into that process, right? Which means we're trying to put things together. That's going to be an anabolic process, right? And for the most part, right, the universe doesn't want to be put together. It wants to fall apart. Let's see here. This particular equation for photosynthesis that we have, is it inorganic or exergonic? It is, because why? How can we tell? Delta G is positive, right? That means it's inorganic. That means it's catabolic or anabolic. Anabolic, which we already knew because it's photosynthesis, right? Spontaneous or non-spontaneous? Non-spontaneous, right? Those are all true statements. Let's see here. Good. All right, what is this molecule? We already reviewed this before the break, but let's review it again. What's this one? This molecule that you should recognize immediately without thought. You should just look at it and instantaneously know that it's ATP. One big clue is it has 3 freaking phosphates on it, y'all. Right? The other clue is it has ribose with a nitrogenous base, right? This is adenine together with ribosome is adenosine. And when we attach 3 phosphates to adenosine, we call that adenosine triphosphate, which is what ATP stands for. The adenosine is the adenine, right, plus the ribose. These two together are known as adenosine. Right? If this was guanine attached to ribose, that would be guanosine. If this were just lost it, just went out of my head, right? Cytosine, actually it would still be called cytosine. And then what's the other one? I did cover this before the break. Thymine, yeah. And then it would be thymosine, right? So yes, right, adenosine, right, one of the bases for DNA, but when we attach 3 phosphates to it in this particular configuration, we have ATP. What is this molecule that you should instantly recognize? What's that? No. It's a radically different molecule. You can't call it the same thing, it looks different. It has similarities, you're right. This is going to be a 105-point question on the 100-point exam on Thursday.
Oh, yeah.
If you get this one question right, you don't have to answer any of the other questions. It does have a similar structure, right? We see, right, that we have the adenosine still. Right? This adenosine has a couple of phosphates plus an extra one down here. Right? But then we have this little bit here. And then we have, here's your big clue right there, the sulfur on the end. What have we seen in cellular respiration particularly that has a sulfur stuck to it? Sometimes just see it. It's coenzyme A. That is coenzyme A, right? Adenosine plus a B vitamin plus a cysteine, which is one of the sulfur-containing amino acids. The liver puts those three things together, and bingo bango, you have coenzyme A, otherwise known as CoA. Where do we see CoA pop up in cellular respiration? Near the end. Not really. Near the end of glycolysis, sure. Not near the end of cellular respiration. The whole process. It is, right, attached to what to facilitate its entry into the mitochondria? What do we get at the end of glycolysis? ATP is one of the products, but what is the molecule? What is the remnant molecule of that glucose that we started with? Pyruvate. And then pyruvate is oxidized to acetyl-CoA. I think I almost heard somebody say it. Yes. Right? The pyruvate loses one carbon as carbon dioxide. It's now a 2-carbon molecule, which is an acetyl group, and it has that coenzyme A stuck to it. The coenzyme A is what facilitates the entry of that acetyl group into the mitochondria so that it can participate in the citric acid cycle. By combining with what? The first step of the citric acid cycle is to combine that acetyl-CoA with oxaloacetate to create the namesake of the cycle, citrate. Very good. All right, we also see CoA pop up again. You get succinyl-CoA. Right, about halfway around the Krebs cycle or the citric acid cycle. All right, what is this molecule that you should instantly recognize?
NADH.
That is almost right.
NAD+.
If I've been pointing to this one, you'd be correct. But I was pointing to this one. This one is NAD+. This one is NADH. It's got the extra hydrogen. What is the point of NAD+? What's its purpose in life? It's ikigai, if you will. You don't know what ikigai is? Does everybody know what ikigai is? It's Japanese for your purpose in life. Yeah, what's the purpose of NAD+? What's the point of it?
Contributes to reactions.
Very general sense, I suppose you are correct, yes. What is the specific purpose of NAD+? What's that? Yes, it's an electron carrier, right? It chaperones those electrons, right, from the cytosol where glycolysis just happened to the mitochondria where the citric acid cycle will happen. We will generate more NADH in the citric acid cycle. Right? That's just more electrons. And ultimately those electrons are destined for where? Or what process? The electron transport chain, right? That's where those electrons are going to go. Right? What similar molecule serves the same purpose? FAD, which picks up 2 electrons to become FADH2, right? If you want to see a picture of that one, that was in some slides that I presented before the break. All right, so being able to recognize an ATP molecule. I'm not sure we're there yet, but you're going to get there, right? Recognize its components, right? What do I mean by its components? I mean the adenine, the ribose that make the adenosine, plus the 3 phosphates. Right? How does ATP actually power cellular processes? In a few words, right? What is actually happening? What's that? It does, right? But how does that power a process? How is that helpful? And leads to what? You're just saying things, Manish. Yeah, what happens when we transfer that phosphate to the molecule that now has a phosphate stuck to it? So what? It has a phosphate stuck to it. It is phosphorylated by attaching a phosphate to it, right? But it's not like a little engine. It's not like we attached a rocket to it and now it's rocketing around, right? What is the consequence of attaching a phosphate to another molecule? What happens? This is like a foundational key concept that will serve you for the rest of your life, so I'm going to write it in red super big. There we go. Conformational change, right? It changes shape, right? Phosphate has a negative charge. So if you stick a phosphate to a protein, which has lots of positive and negative and polar groups and things like that, it's probably going to change its shape. And we call that conformational change, right? And that change in shape, right, is what facilitates something happening, right? So when we power a reaction, right? So for example, those proteins that I showed you, right, that have that big vesicle behind them and they're dragging them down a microtubule, they're walking down that microtubule, right? What makes the legs change shape so they can walk down that microtubule, right? Attaching an ATP and then detaching it, right? Muscle contractions, right? Thick filaments and thin filaments have to grab onto each other and ratchet and let go and grab and ratchet and let go and grab and ratchet and let go. What powers that? ATP through conformational change, right? That's how ATP actually does what it does, right? Change in shape. Let's see here. Activation energy. What does that even mean? Why would we need there to be activation energy? What are we trying to overcome? Sometimes, yeah. Right. It's that shift in shape, that bond that needs to be changed, right? Induced fit, right? Sometimes we bind a substrate, right? And when that substrate binds, it's kind of the same thing as when ATP binds, right? It's going to cause a shift in the shape of the enzyme, for example, so that it fits that substrate, right? To create that microenvironment, right? Change in pH, perhaps, change in electronegativity, change in pH. Did I say that already? Right? These things, right, all lead to need to be some sort of barrier to get over. Right? And an enzyme can help facilitate that. How does a cofactor involved in that process? What is a cofactor? Can anybody name an example of a cofactor? Maybe something that we've already talked about that has the word co as part of its name. Coenzyme A is an example of a cofactor. Who's got another? Does hemoglobin work without iron? Iron is a cofactor. And hemoglobin is worthless if it doesn't have iron, because iron is what actually binds the oxygen. Okay? Acetyl-CoA, right? Acetyl can't enter the mitochondria without that coenzyme A attached to it. Right? That facilitates the process. Right? Cofactors, right, are pieces, keys if you want. Right? You can call it a key that opens the lock, right, that fits into something. The way it's often represented in a textbook is you have an enzyme, right, and you have a substrate. It looks like that, right, and it won't bind to that pocket until you get a cofactor in there, right, to help it fit that shape, right, because the substrate wasn't the same shape as the pocket there until we put our cofactor there. So the cofactor, right, makes the binding pocket the right shape so your substrate can fit in there. Right? Some cofactors are things like metal ions, like iron. Right? If the cofactor is another protein, right, then it's called a coenzyme.
So are these molecules cofactors?
Which molecules?
Like everything?
No, ATP is not a cofactor because it doesn't fit into something to make it complete. Coenzyme A is, right, a cofactor. NADH is just an electron transporter, right, so it doesn't fit into another enzyme, right, to give it functionality. Right. A cofactor has to attach to an enzyme or attach to another protein in order to make that protein or that enzyme functional.
But doesn't MTP like attach and then temporarily and then detach? Just it's a repeating process.
It does, but it doesn't have anything to do with the binding pocket or the active site. Right? It's not required. I see what you're getting at, right? It's not quite the same thing, right? It's not the same as it's like if you have a motor, then yes, you have to turn the motor on, right? And attaching ATP is what turns the motor on, right? But if your motor doesn't have, I don't know, a cylinder, right, then it's an incomplete engine. It could not be turned on anyway, right? And an enzyme can be the same way, right? Attaching a phosphate, right, is what powers that enzyme to do what it does, to conformational change, right? But if the enzyme itself is not complete, it doesn't matter how many ATPs you attach to it, it's never going to do its thing because it's not complete yet, right? So maybe rather than just saying attaches to it, right, makes it complete. Inhibition. Right, there are two types of inhibitors. What are they? Competitive and non-competitive. What's the difference? Yeah, very good, right? A competitive inhibitor, right, competes, right, with whatever's supposed to bind there, right? That's why it's called competitive, and so it can bind the active site. Right? Non-competitive is not even competing for that active site. It doesn't care. It's going to bind somewhere totally different, but that'll still cause a conformational change, right, which will make the binding pocket no longer, right, the active site, and so that's called allosteric. Very good. Let's see here. What are some ways that enzyme activity can be regulated in cells? Because we don't want enzymes active all the time, right? Otherwise, they would just be doing everything all the time. We'd be building things and tearing things down all at the same time, right? So what are some ways that we regulate whether an enzyme is on or off? Yeah, and what might that inhibitor be? Coffee's a stimulant. And unrelated to this, because we're not talking about exogenous substances, we're talking about endogenous substances. Yeah, the product of a reaction, right? So very often, right, if you have, right, a cellular process, right, one enzyme to the next, the next, the next, the next, right, the thing at the very end of the chain, right, whatever the product of that series of reactions is, right, will feed back. That's why it's called feedback inhibition, right? The product itself will inhibit the enzyme that started the process in the first place, right? This is how, right, we make sure that we turn off the system once we have enough product, right? If you have a bunch of that product, you don't need to make more of that product, so the product itself can act as an inhibitor. That's one way that we can regulate enzyme activity. What's another one? That's a type of allosteric inhibition. Yeah. Negative feedback is almost always allosteric. Right? It's not going to compete for the binding site, right? Instead of binding somewhere else in the allosteric site. Compartmentalization is another good one. Right? Compartmentalization just means that we keep the enzymes somewhere else. Right? So for example, right, not all of cellular respiration happens in the mitochondria. Right? Some of it, the glycolysis part, happens outside the mitochondria. Citric acid cycle and electron transport chain or oxidative phosphorylation are inside the mitochondria. Right? So those enzymes are separated from each other, right, by just being compartmentalized. A lysosome, right, has enzymes that would lyse all kinds of proteins inside the cell and tear that cell up unless they were compartmentalized. Right? Those enzymes are kept inside the lysosome. Right? Peroxisomes, right, also have peroxidases in them. We work with peroxidase. You probably don't want peroxidase running rampant inside your cell. Right? We keep peroxidase inside a peroxisome so that it's only active on the things that we want it to be active on. That's another way that we can regulate enzyme activity, right, is by sequestering it, right, or compartmentalizing it away from stuff we don't want it to act on. Another big one, phosphorylation. Right? We turn enzymes on by phosphorylating them typically. We turn enzymes back off by dephosphorylating them. Right? So the very fact that enzymes can be turned on and turned off by phosphorylation, dephosphorylation, is another way that we regulate enzyme activity. Those are 3 big ones right there. All right, let's distinguish between aerobic respiration, which requires what? Specifically, oxygen, yeah, right? Aerobic respiration is called that because it requires oxygen. Anaerobic respiration does not require oxygen, right? How about fermentation? Oxygen, yes or no? It's another anaerobic process. Glycolysis, aerobic or anaerobic? Just glycolysis. Yeah, just glycolysis is anaerobic, right? Even though it's part of an overall aerobic process, cellular respiration. Cellular respiration as a whole, aerobic process. Just the glycolysis part, anaerobic. They are all redox reactions, which means what? One thing is, yeah, electrons, right? So electrons are transferred, right, from something which is oxidized to something else which is reduced. All right, let's make sure we can identify that for each of these. For aerobic respiration, otherwise known as cellular respiration, what is it that's oxidized at the very beginning of that process? What was that? Glucose, yeah. What is reduced? Oxygen isn't reduced. Kind of a tricky question. There's not really a very good answer. You might say pyruvate, right, because that's where the electrons come from. Right? You might say, that's probably what I would say actually. What's the final electron acceptor in the aerobic respiration process? It is oxygen, right? That's where those electrons ultimately go. What about anaerobic respiration? What is it that's oxidized? It's still respiration, y'all. It's not that different. It's still glucose, right? What is reduced? Again, you might say pyruvate, right? But in this case, what is the final electron acceptor? Yeah, could be a number of things, right? Might be sulfate, might be nitrate, right? Could be even carbon dioxide, right? Something like that, right? There are a few different possibilities, right? The point is not oxygen. What about fermentation? What's oxidized? Still glucose, right? What's reduced? What's the final electron acceptor? Pyruvate again. Pyruvate gets those electrons back from NADH. Right? NADH has those electrons. Right? NADH gives those electrons back. Okay, 2 types of fermentation. What are they? Bless you. 2 types of fermentation. Alcohol fermentation is one of them, yeah. And lactate, or lactic acid fermentation. Which one do humans do? Very good. Let's see here. Oh yes. Why would fermentation happen in a human cell? Lack of oxygen, right? So what's a good example cell type where fermentation frequently happens? Muscle cells. Right? Muscle cells are one of the most common cell types in the human body to experience fermentation on a regular basis, right? Because they just are doing more, right, than they can with the amount of oxygen available to them. And so when they run out of the ability, right, to use oxygen as that final electron acceptor, they have to start shunting, right, that pyruvate somewhere else, and they shunt it down the fermentation process instead. What happens, right, so after that pyruvate, right, is oxidized, right, it's turned into lactic acid, it's released into the bloodstream, and then what? Where does it go from there? Pick an organ. The liver. And what does the liver do with it? You say it turns it back into pyruvate? And then what does the liver do with it? Oxidizes it again, but this time to acetyl-CoA, and into the mitochondria to complete cellular respiration. Another cell type that frequently does fermentation is red blood cells, simply because they don't have mitochondria. Here are those two processes. Okay, so we start out with glucose. As we work our way down to pyruvate, we generate NADH. Right? It's a redox reaction. We're taking those electrons, right? We're carrying them on NADH. Right? Then the pyruvate, right? If we can't do anything with it, right, becomes ethanol. Right? So that NADH, right, can give those electrons back, and we regenerate our NAD plus, so that we have NAD plus to use again. That's the whole point of the process, right? Because we can't just let pyruvate build up, right, without doing anything else, because eventually we'll run out of NAD plus to hold those electrons, right? And so then glycolysis stops too. And if we're not doing cellular respiration because we don't have oxygen, and we're not doing glycolysis because we ran out of NAD plus, then we're just not doing anything, right? And so this process, right, at least regenerates the NAD plus so that we can continue to do glycolysis. We still can't do the rest of cellular respiration, but we can at least do that. Right, those electrons are given back to pyruvate to create ethanol in alcohol fermentation. Right? Same thing here, glucose to pyruvate, right, generates NADH. Right? Pyruvate gets those electrons back from NADH, thereby giving us our NAD plus, so we can keep on doing glycolysis, right? And we have lactic acid as the result. Two common types of fermentation. This one over here, the one that humans do, just so we can keep glycolysis going. All right, what about this? Right, basic steps of glycolysis. Who is prepared to draw glycolysis from memory? It's okay, you don't have to. But you do need to recognize what's happening at each step, right? So the types of enzymes. Right, do we know what kind of enzymes involved in the first step? What is the first step? What happens in the first step? You don't even have to know it off the top of your head. You can just look at the picture and tell me. Yeah, it gets phosphorylated, right? Here we have glucose. This glucose is exactly the same, except it's got a phosphate stuck to it, right? That's just there for you to see, right? Nothing you have to memorize there. You can just look at it and see that, right? If it's phosphorylated, then what kind of enzyme must have been involved? A kinase. There we go. So that's the type of enzyme. Right? In the second step, right, from here to here, what is it that happened? Right, here we have a 6-sided ring. Here we have a 5-sided ring. Everything else is the same, including the phosphate. What do we call a 5-sided sugar? In this particular case, it's fructose. Right. So. Right? Glucose and fructose, same molecule, just put together differently. So what kind of enzyme must have done that? An isomerase, because they're isomers of each other. Third step, what's the difference between this one, right, fructose 1-phosphate, and fructose 1,3-bisphosphate? There's an extra phosphate, so what enzyme must have done that? You guessed it, another kinase, right? And so on and so forth. That's the sort of thing that I'll expect you to be able to do on the exam, which is in 2 days, right? Just look at the step, right? Recognize what happened, right? And then infer what enzyme must have been involved based on what you see that happens there. It's just a matter of looking at it and noticing the differences. If you look at it 4 or 5 times, right, you should be able to recognize what has happened in each one of those steps. At which 2 steps did we use up an ATP? I got to spend money to make money. There's an energy investment phase, and there's an energy, what do you call that? Reaping phase. There's a different word for it. Which 2 steps did we use up an ATP? Just shout out the numbers. If it helps, they're colored purple. 1 and 3, very good. Those are the steps where an ATP was used. How do you think you might have been able to figure out that without knowing it? Well, guess what? A phosphate suddenly appeared. So it seems like that phosphate would have come from somewhere, probably an ATP. In this step, we already talked about it, another phosphate appears out of nowhere. No, not from nowhere, from an ATP, right? So again, if you just look at what happened, right, and you see a phosphate appearing, probably came from ATP. There is one case where that's not true. Which step is that? Which step? Did you say 6? Just nod your head yes. Put one finger down and say yes, 6. Yeah, very good, Louise. Yes, in step 6, right, we got a phosphate added, but it didn't come from ATP, right? Instead, right, the GAP-DH enzyme, right, took inorganic phosphate. Remember inorganic phosphate? Right, there's also phosphate that just exists in the cytosol, and some enzymes, right, like this dehydrogenase, right, has the ability to use that inorganic phosphate. Let's see here. Where was an ATP generated? Which 2 steps do we generate ATP? Yeah, they're colored yellow for you. They won't be on the exam, right? But again, right, we've got 2 phosphates, and then suddenly we only have 1. Right? We have 1 phosphate, and then we have none. Right? And so if we're losing phosphates, right, then that's a good clue that it's because we're regenerating ATP. Let's see here. Carbons and electrons. We know where the electrons go, right? Where do they go? We just pull the electrons off of these molecules and what, do we hold them in a basket? Basket, that's the answer? Yeah. Put them in our back pockets. Put them in a lysosome. We transport them in the mitochondria. They're just free floating out there. They find their way, they're very independent, they can do it by themselves. Or maybe we have a chaperone called NAD, FAD, right? That's where the electrons go. They go to our chaperones, NAD and FAD. Where does all of this happen inside the cell? Mitochondria? Lysosome? Endoplasmic reticulum? The nucleus? Come on, where's it happen? In the cytosol, right? No particular organelle, right? Glycolysis just happens in the cytosol. If you'll recall, it's all crap. Whatever. You have a cell. We have an insulin receptor. We have insulin. Okay? When insulin stimulates the insulin receptor, it goes into the cell. It finds a glucose transporter called GLUT, right, which translocates to the surface. Right? Then our glucose, right, can go through the glucose transporter into the cell, where it's phosphorylated. Right? And then glycolysis continues from there, right? This is just in the cytosol. Right? No particular organelle. Once that glucose goes through the glucose transporter into the interior of the cell, it's just inside the cell, right, in the cytosol. Right? Then, right, 10 steps. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 steps. Right? We get pyruvate. Right? Pyruvate becomes acetyl-CoA. Then the acetyl-CoA goes into the mitochondria where the citric acid cycle and oxidative phosphorylation happen. Right, so glycolysis in the cytosol, right, citric acid cycle and oxidative phosphorylation in the mitochondria. Let's see here. What about fat metabolism? Where does that intersect with all of this? Right, there's a fatty acid, a little simplistic. You know, it's got some other parts. Here's your fatty acid. Fatty acids also transported in the mitochondria, where we chop them up through a process called beta oxidation into multiple 2-carbon groups called acetyl. To each of those acetyl groups, we attach a coenzyme A. And so this fatty acid, right, gets converted into 1, 2, 3, 4, 5, 6 acetyl-CoA to join all the acetyl-CoA that came from glucose, which also got transported in the mitochondria, right? So those two pathways intersect in the mitochondria, right? A fatty acid is nothing more than a precursor to a whole big pile of acetyl-CoA. Right? Glucose, right, is nothing more than, after we break it down, a couple of acetyl-CoAs. Right, so inside the mitochondria doesn't know the difference. Right? It just knows that it has a bunch of acetyl-CoA coming from both fats as well as carbohydrates. Right? So that's where fatty acid metabolism, right, intersects. What about protein metabolism? If we're going to break down a protein, which we don't do very much of, it's kind of a waste of what? Why don't we burn proteins? Number 14 on the periodic table. The air is 80% nitrogen, right? Amino acids, right, have that all-important amino group, which is a nitrogen, right? Carbohydrates and fatty acids don't have those, right? It's a huge effort, right, to get nitrogen out of the air through nitrogen-fixing bacteria into the soil as nitrites and nitrates, right, where plants then can soak them up and make use of them. And when we eat those plants and we get that, right, just to turn around and burn it, come on, that would be a huge waste of resources. Question?
Just a clarification, it's number 7, not number 14.
Yeah, that's what I meant. Half of 14. That's what I said. I said it's number 14 divided by 2 on the periodic table. You just didn't hear me say it because it's noisy. What am I thinking of 14 then?
Yes. You was talking about how it's like similar.
Wait, what's the molecular weight of nitrogen now?
It is 14.
Oh yeah, that's what I was thinking of. There we go. I knew it was 14 one way or another. It's just not number 14. I'm not a chemist. Let's see here.
Yeah.
Yes.
So is that acetyl-CoA and it goes into the mitochondria through NADH? Like NADH is the carrier that takes it to the mitochondria?
No, coenzyme A is the carrier.
Oh,
It gets the acetyl group into the mitochondria.
Does NADH play a part in that? Or
Not that process, no. NADH is off doing its own thing, carrying those electrons in there as well. And off the top of my head, I'm not immediately sure how NADH actually gets into the mitochondria. But I'm sure there's a transporter for it.
So coenzyme A attaches to pyruvate, or how does pyruvate turn into the acetyl-CoA?
So you have a dehydrogenase, another enzyme, right, that oxidizes the pyruvate, right, basically takes off one of the carbon groups, which leaves the CO2, and puts in its place, right, that coenzyme A. Right, so there's one enzyme that does both of those things. Right? It removes, because pyruvate's 3 carbons, right? So the same enzyme, right, removes that 3rd carbon and attaches that coenzyme A in its place. That's all one step. That's step number 11 if you want. So we have 10 steps to pyruvate.
It's not on there.
It's not that, yeah. 10 steps to pyruvate, glycolysis is done, because that's where we have the split in the pathway where we can either go down the fermentation pathway, right, or we can continue down the cellular respiration pathway. Right? If it continues down the cellular respiration pathway, right, then that pyruvate is oxidized to acetyl-CoA. Two questions. Let's see here. Speaking of the citric acid cycle, we already talked about our acetyl-CoA. Here it is. Right, so there's the 2 carbons of acetyl-CoA. Right, the CoA's done its job, so we don't need it anymore. We attach those 2 carbons to the 4-carbon oxaloacetate to get citrate. That's our 6 carbons. Right? What do you think happens at A there? Not yet. Not ATP, not yet. Right, we've gone from 6 carbons to 5 carbons, so where did that carbon go? I'll show you. CO2, right? So citrate to isocitrate, right? And then on down and around. I don't actually care if you know the names of those, right? But I do want you to recognize that we lost a carbon there. Right? That leaves us carbon dioxide. And in that process, we pick up another NADH. 5 carbons to 4 carbons, so at step B, what happens? Another carbon dioxide, another NADH. Right? From here, right, 4 carbons, we still have 4 carbons here, so we're not going to lose any more carbons the rest of the cycle. Right? We lost our 2 carbons here at A and B, right? We do that right off the bat, right? Carbon dioxide, NADH, NADH. Right? Step C, right? Does anybody know what we get there? There's all those people that were eager to say ATP before. Yeah, except it's GTP. Very similar, right? It's just like ATP, but it's GTP, right? So that's what we generate there, right? What about D? It's actually another NADH. What about E? FADH2. Yeah. The citric acid cycle doesn't really generate very much energy. We get that 1 GTP and that's it, right? But we get 1, 2, 3 NADH plus an FADH2. All right, we already talked about this one, right? But let's just make sure we recognize it when we see it. Right? What do we see here?
Shapes.
Hopefully you see more than that. Maybe what's represented by the shapes. What do we see happening here, right? Substrate binds the enzyme, right? Enzyme turns it into product. Product feeds back and binds not in the active site, but somewhere allosterically, right? Changing the shape of that active site so that it's no longer active. It can't bind that substrate, right? That's negative feedback, right? That's that feedback control that we were talking about. This one specifically is negative feedback where the product, right, inhibits the enzyme that led to the creation of that product in the first place. Can anybody think of an example, a specific example of this in cellular respiration? We have glycolysis, right? Glucose to pyruvate, right? Then acetyl-CoA, then into the citric acid cycle, right? Citrate is the first thing created from the citric acid cycle. If you're getting a lot of citrate, that must mean that you've broken down a lot of glucose, and so you probably can slow it up, and so citrate actually inhibits the very first enzyme in the process, right? Phosphofructokinase, sorry, the third enzyme in the process, right? Phosphofructokinase is the one that phosphorylates that fructose, right, that we got after the isomerase, right? And so the citrate that's created down here, right, inhibits, right, more coming down the pathway. If citrate starts to get low, that's an indication that we need to restart this cycle and generate more of it. So as citrate gets low, less of it is bound to phosphofructokinase, which means it starts to become active again. This becomes active again, and we start feeding it in there again. So citrate, in this case, right, feeds back on the third enzyme of the glycolysis pathway. ATP itself, right, can also inhibit phosphofructokinase. Right? So if we're generating a little bit of energy from oxidative phosphorylation, right, again, that's a signal to the cell that we've done a good job. Right? We've fed things down the pathway, we've generated our energy, and we don't need any more of that. Here's our ANP. Do we remember what ANP is? It's just like ATP, except only one. It's It's adenosine monophosphate. A lot of times adenosine monophosphate wraps back on itself and becomes cyclic AMP. Right? Cyclic AMP, right, is one of those second messengers that we talked about in the previous unit. Right? And that one can stimulate it, right? Because if we have lots of AMP, that means we've used up all of our ATP, and now we need to restart that process, right? So that can stimulate the enzyme. Two examples, right? One, right, stimulating, one inhibiting.
Is the substrate a good example, like of being a cofactor, or no, not really?
No, the substrate is what the enzyme acts on.
Oh, okay.
It's not necessary for the enzyme to be complete, right? It's the actual thing that the enzyme works on. I see how you think that though, because it's fitting into a pocket, right? But that pocket was made for that substrate. All right, let's turn our attention to photosynthesis. What's an example of an autotroph? A plant. Why is a plant an autotroph? Because it can make its own food, right? Auto, right? What about heterotrophs? What's an example of a heterotroph? Anything that eats something else, right? The consumers, right? If you want to think of autotrophs as the producers, heterotrophs as the consumers. Let's see here. You want to go any further into that? I don't think so. What are these yellow cells up here? I have a very similar covering on my body. Even the one that I was born with, not the surgical one. Yeah, it's the epidermis. All right, we have an upper epidermis and we have a lower epidermis. What's this hole here called? This hole through which oxygen enters the leaf and carbon dioxide leaves the leaf. Well, just one hole would be a stoma. Multiple stoma would be stomata. The two cells that regulate the opening and closing of this stoma, what are they called? They guard the entrance to the leaf if you want, so they're called guard cells. Very good. What about these dark green cells along the top here? What are they? Type of mesophyll. Which type of mesophyll? Palisade or spongy? Palisade, you have a 50-50 shot, yeah. These are the palisade mesophyll cells, right? These are the spongy mesophyll cells. I forgot to put a chloroplast up there. Both chloroplast and mitochondria were once thought to have been separate organisms which were enveloped by eukaryotic cells through a process known as what? Or what is the theory called? Yeah, endosymbiont, right? The symbiont that lives inside, right? Endosymbiont. So a chloroplast has how many membranes? It does, just like the mitochondria, right? And then inside the chloroplast, we have additional structures, which are called thylakoids, right? How many membranes does a thylakoid have? Just the one. Trick question. And embedded in the thylakoid membrane or the chloroplast membrane is where we find all the photosynthesis proteins. They are in the thylakoid membrane, and they are surrounded by stroma. Yeah, stroma, not stoma, right? Stroma with an R, right, is the fluid interior, right, of a chloroplast. All right, that's what we're looking at, right? So up here, right, we have the fluid-filled interior of the chloroplast, which is called stroma. Right, here we have the single membrane, right, of the thylakoid. Right, this is the thylakoid lumen, or the inside of the thylakoid. right? This is called what? Photosystem 2, right? Remember, photosystem 2 comes first. I know, I'm sorry. Right, then we have PQ. What's PQ stand for? Plastoquinone. Very good. Then what's this next one? CYT stands for cytochrome. Right, cytochrome C. Then we have PC. Plastocyanin. Right, followed by photosystem 1. Right, FD. Ferredoxin. Right, and then finally, right, we have our reductase. Right? What's the difference between NADP plus and NAD plus? NAD plus and NADP plus. What's that extra P?
Phosphate.
It's an extra phosphate, right? The only difference between NAD and NADP is NADP has an extra phosphate, right? What's the functional difference between the two of them? There's no functional difference. They are, for all intents and purposes, the same molecule. Just like GTP, right, is an ATP analog, NADP is an NAD analog, right? They have the same function. You can view them as the same. What are all the little green dots on the photosystems? Come on now, it's a plant, it's green. What do you think it is? It's chlorophyll, not chloroplast, right? We're inside the chloroplast right now, right? We're deep inside the chloroplast, right? They're chlorophyll molecules, right? What do we have right here, right? These two chlorophyll molecules right next to each other. They're special somehow. How are they special? What do we call that area? It's the reaction center. Right? The reaction center, what happens there? What happens when light hits a chlorophyll molecule in the first place? It excites the electrons, right? When that energy, right, exciting those electrons, right, bounces from chlorophyll to chlorophyll, it finally gets to these two special chlorophylls, right? When those electrons are excited to their, right, excited state, right, do they just fall back down to their ground state like they do with the others, or does something else happen? Yeah, they're actually pulled away, right? They actually lose those electrons altogether, right? So electrons, right, excited, right, as they become unexcited, that energy transfers to another one, excites them, right, they fall back, excite in another one, and so forth, on and so forth, until finally they get here. But the difference here is when they get excited, they're near, right, the primary electron acceptor, right, and so rather than being excited and then falling back to their ground state and passing that energy off to another chlorophyll, they're excited, and then they're actually stolen away, right, by the primary electron, or by the primary electron acceptor. Okay. How do we replace those electrons then? Right, these two special chlorophylls in the reaction center just lost their electrons, right? So how do we get them back? From water, right? So we split water. It's happening right here, right? Splitting water produces oxygen. That's why plants, right, produce oxygen as a byproduct, right? Also a couple protons, and those electrons replace the electrons that were just stolen away there. From there, right, we have the electron transport chain, right? So from there, right, so plastoquinone to cytochrome C to plastocyanin, right? Then the same process here in number 1, photosystem 1, then the ferredoxin, right? And finally, those electrons, right, go on to NADP, right? Because it is, after all, an electron chaperone. At the same time, right, what do we see happening right here? Protons, they're not a full hydrogen, right? But plastoquinone and cytochrome C, right? Both are going to use that electron as they pass it along the electron transport chain, right? They're using the negative charge of those electrons to draw the positively charged protons down from the stroma and into the thylakoid, because that's the name of the game, to concentrate protons. Doesn't matter whether it's cellular respiration inside the mitochondria or photosynthesis right here in the thylakoid. Right? The name of the game is to concentrate protons, and that's why we're gathering up all those electrons in the first place. None of the rest of it matters. Right? All the rest of it, right, is just the game we play to get those electrons so we can concentrate that protons. Everything else is just extra. Right? It's the process that had to happen for that to happen. Right? So all those protons we concentrated, right, that represents potential, right, chemical energy, right, and then they're going to flow down their concentration gradient back into the stroma, and that energy is going to be used to generate ATP. That's how we get ATP. Where's that NADPH where those electrons going to go? Calvin cycle, y'all. It's going to go into the Calvin cycle, which is right here. Let's see here. Well, I might as well point out, right? Rubisco, what does Rubisco stand for? There's a couple of clues. Right, so Rubisco is the enzyme, right, that attaches or fixes carbon dioxide to ribulose bisphosphate. So what do you think Rubisco stands for if it's doing something with ribulose bisphosphate? Ribulose bisphosphate carboxylase. Right? Because it takes ribulose bisphosphate, that's the first part of its name, right? And it combines it with carbon dioxide, that's what a carboxylase does. Right? So the very name of the enzyme is exactly what it does. Right? It attaches carbon dioxides to ribulose bisphosphates. Right? This creates a couple of intermediates, right? And eventually, right, we get to glyceraldehyde 3-phosphate. Where have we seen glyceraldehyde 3-phosphate before? Where have we seen that though? G3P, we've seen it once before. It's an intermediate in cellular respiration too. Same molecule, two places. And then, right, this glyceraldehyde 3-phosphate is going to be used to make what? Glucose. Right? That's why it appears in two places, right? Glyceraldehyde 3-phosphate is used, right? 3 carbons plus another 3 carbons, that gives you 6 carbons, that's glucose. Right? So it makes sense as we break down that glucose, we would get glyceraldehyde 3-phosphate, right, somewhere in that process as well, right? Glyceraldehyde 3-phosphate can use to build glucose, or it can break glucose down to glyceraldehyde 3-phosphates, and then further to pyruvates. Right? And then the rest of this just regenerates the ribulose bisphosphate, very much like the rest of the citric acid cycle, regenerates oxaloacetate.
So, glycolysis is linked to the citric acid cycle, and the electron transport chain is linked to the Calvin cycle. Is that what it is?
Well, there are 2 electron transport chains. Right? So after the citric acid cycle, right, oxidative phosphorylation is an electron transport chain. But now we're talking about photosynthesis,
Yeah.
right, where we start with an electron transport chain rather than ending with an electron transport chain.
Oh, okay.
2 separate processes.
Oh, yeah.
Right? They just have similar aspects to them. Right?
Yeah.
So both cellular respiration and photosynthesis have a electron transport chain. Right? Both of them, right, use NAD or NADP, transport electrons. Right? Both of them, right, have glyceraldehyde 3-phosphate as an intermediate, right, either on the way to building something up or on the way to breaking it back down. Right? So there are a lot of similarities between the processes because they're essentially the reverse of each other. Let's see here. there anything here I haven't already talked about? I don't think so. This is just a closer look, right, at one of the photosystems. I already talked about the photosystem, but here's a kind of a blown up version of it. Right, so again, right, photons, right, exciting electrons, right, that energy kind of bounces around till it gets here. Right, the electrons are liberated, right, to the primary electron acceptor, right, in the reaction center, right, and then it gets passed on, right, to plastoquinone, cytochrome C, et cetera, et cetera. So nothing here that we didn't see in the previous diagram. Let's see here. What are we looking at here? Same kind of thing. What's the difference between this image and this image? Similar processes, but there's a key difference. What's that? Well, the second one, sure. Right, and this one, right, so C4 photosynthesis, remember, is an adaptation that plants living in hot, arid environments will utilize. Right? For one reason, right, if you're in a hot, arid environment, you probably have less water available, and we need that water to get those electrons for the electron transport chain. Right? So if we don't have enough water, we can't really do that. Right? Also, when it's hot, right, plants tend to try to conserve water by closing their stomata. Right? If they close their stomata, they're not getting fresh CO2 for the Calvin cycle, but they are still generating oxygen. And remember, Rubisco has an equal ability, right, to fix carbon dioxide and oxygen, right? But when it fixes oxygen, right, you're not getting the carbon, and the carbon's the whole point of carbon fixation, right? And so it's just kind of a waste. It's called photorespiration, right? When Rubisco, right, has high concentrations of oxygen available to it, it will just fix that oxygen to ribulose bisphosphate instead, right? And that's just a waste of resources. So plants, C4 plants, avoid this, right, by making their carbon dioxide physical, right? So that's what's happening here, right? We have the CO2, right? It's coming into our mesophyll cells. It's being fixed as a C4 acid. Does anybody remember what that 4-carbon acid is called? I covered it real briefly. It's called malate, right? But that's why it's called C4 photosynthesis, right? Is because they take phosphoenol pyruvate, PEP, right? We also saw phosphoenol pyruvate before in cellular respiration. Let me just remind you. Right? The next to last step is phosphoenol pyruvate to pyruvate. Right? Same molecule that we see as we break down glucose. Right? Used by C4 plants right here. Right? So that phosphoenol pyruvate, right, can be combined with carbon dioxide to create malic acid, which is a 4-carbon compound. That's why it's called C4 photosynthesis. Right? And then it's transported and stored right here in these bundle sheath cells. So remember, C4 plants, they don't have spongy mesophyll. Instead, they have a modified spongy mesophyll, which is called a bundle sheath. And the point of the bundle sheath cells is to store carbon dioxide as an organic acid, right, as malates. That way, right, when the plant starts to increase levels of oxygen, right, and it's running out of carbon dioxide because it had its stomata closed, it's okay, because it has an extra storage, right, of carbon dioxide, which is combined, right, as an organic acid, right? And so when the stomata are closed and it's not getting fresh CO2, then it can liberate CO2 from that malate, right, to be used, right, for the rest of photosynthesis. That's an adaptation that plants living in hot, arid environments have made. This is a spatial adaptation, right, because they've taken CO2 out of the air and they've removed it to a different cell to store it. Here, right, we have a variant of C4 photosynthesis called CAM, C-A-M. I still don't remember what CAM stands for. I won't ask you to remember either. Something like cerulean acid.
It's actually
Yeah, that, right? Right, so similar process, right? Except that they don't have two different cells. It all takes place in just the regular mesophyll cells, right? But it's a temporal separation, right? During the day, they close their stomata, and at night they open them, right? So at night they get fresh CO2, and they store all that CO2 as malate, right? So that during the day, when their stomata are closed, they can liberate the CO2 from that malate to use it again. Right? The only difference, right, is how they go about doing it, right? Regular C4 plants store all that malate in a separate cell called a bundle sheath cell, right? Because, right, sometimes their stomata are open, sometimes they're closed. It just depends on how hot and how dry it is, right? If it's a really wet, cool day, right, then they'll just do photosynthesis as normal, right? But if it gets hot, right, and it gets a little bit drier, right, then they activate this process. Right? Cam plants, right, are things like succulents, cacti, things like that, right? They don't really ever have wet, cool conditions, right? It's always hot in here. And so, right, they just always do the day. Right? But it's the same thing. Right? We're just storing carbon dioxide as malate, right, so that we can use it when we need it. Oh yes, this is cyclic electron flow. Right? This is relevant to C4 and or CAM metabolism, or photosynthesis rather, right? Because this is how they keep photosynthesis going, even right, when they don't have enough water. Right? So remember, right, to replace the electrons that are stolen away from these chlorophylls in the reaction center, we need a fresh supply of water because that's where we're getting the electrons. Right? But in C4 and CAM plants, they don't necessarily have a lot of water available to them, but they still need those electrons. Right? And since they don't have water to steal the electrons from, they just recycle their electrons. Right? So the electrons, right, from the cytochrome complex go to plastocyanin as normal. They go to photosystem 1. They excite those electrons to the primary electron acceptor. But then, right, instead of passing them off to ferredoxin, right, and on to the NADP so they can go on to the Calvin cycle, right, instead they just pass those electrons back to ferredoxin, back to the cytochrome complex, and they just keep using the same electrons over and over and over again. Right? Why? Why would you want to use the same electrons over and over and over again? What does that get you? What was the cytochrome complex doing with that electron in the first place? Remember, the cytochrome complex was using that electron to draw protons down here, right? The name of the game is to concentrate protons. Right? If we concentrate protons, then we can generate ATP. Right? So in cyclic electron flow, right, since we keep on giving those electrons back to cytochrome, and then giving them back to cytochrome, and then giving them back to cytochrome, it can continue to pull protons down here, which means it continues to concentrate protons, which means they continue to flow through that ATP synthase, and so we still generate ATP. Right? So even though you can't do the rest of the process, right, because your stomata are closed, you don't have enough water, blah, 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 right, you still want to make ATP, right, and so they do that by just reusing the electrons that they have, right, rather than getting new electrons. Right? That's cyclic electron flow, that's related to C4 and CAM photosynthesis. All right, y'all. Seems like we've got a lot of work to do over the next couple of days, but you'll get there. I have high confidence, right, that if you turn your attention to biology for just a few minutes, you can learn this stuff. These slides, by the way, are on Canvas. If you'd like to look through them again on your own.